My name is Dr. Chris Jenkins, and I am the CEO of the Orianne Society and the host of the Snake Talk podcast, the podcast where you learn about nature's most feared, maligned, and persecuted animals. I invite you to listen to this conversation, and maybe you'll find that what you perceive as fear is actually rooted in a deep fascination. Welcome to the Snake Talk podcast. I am here with Dr. Michael Dreslik. I'm really excited about this episode because it really kind of fits into that category of some of my most favorite episodes, meaning Mike or Dres, as he goes by, is a herpetologist that really has, a huge breadth of work across a whole wide variety of snakes and turtles and vertebrates and other animals. So we're going to just have a great conversation today and kind of talk about all kinds of important herpetological research and conservation projects. I first met Dres, I guess, at herp meetings a number of years ago, and I used to go to the herp meetings quite frequently. I'd say in recent years I kind of just go to more regional things, so we really haven't crossed paths too much, but we do share here at Orianne. Our most recent employee came to us from Dres's lab, and I think she came to him from us, so there's this kind of back and forth, which is great. But welcome to the podcast, Dres. How are you doing today?
I'm doing great. Thanks for inviting me.
Yeah. Well, I like to start off, as you probably know, if you've heard some episodes, I like to hear the story. I like the origin story for us weirdos that like snakes and lizards and frogs and these types of animals. Like, how did it happen for you? Was it were you a child? Was it something that hit you later in life? How did this all come to be?
Man, it started off when I was a kid years and years ago. I'm originally from New York and began my early childhood there. I was always interested in dinosaurs and would learn everything I could. My parents would take me to the museum in New York City, and I'd be able to run around and identify the dinosaurs. But active extant herps became my interest when, I know this is a terrible story, my uncle brought me home a box turtle once that was crossing the road. Not saying, this is something that you can do, you should be doing. But it really spurred my interest. From there, it kind of waned a little bit and then picked up again in high school when I was in rural Illinois. I really wanted to kind of be a herpetologist, kid running around catching frogs, catching salamander larvae, and trying to raise them. And that kind of all culminated when talking with Dr. Edward Mall, who is at Eastern Illinois University and a preeminent turtle biologist. I had decided that's the career path I wanted and started my master's degree with him. So my master's work was on turtles. The origin story for the lab, as I tell everybody, it all began with me living in a tent at the side of a lake, to do my master's work. And it has just progressed and steamrolled up until now. Yeah.
That's great. So it sounds like even as when you were in high school and kind of, starting to think about what am I going to do with my life, you had an idea that, being a herpetologist is a job. Like I could do this as a living.
Yep.
Yeah. And so you do your undergrad. I'm assuming you were in Illinois for the undergrad.
Yep. I did my undergrad at Eastern Illinois University too, and volunteered a lot with the graduate students, for their herp projects. And really kind of how I kind of got my foot in the door. A couple of graduate students there, Thomas Wilson and Mike Tuma, kind of really took me under their wing, as an undergrad and helped out.
That's great. And so, with the master's degree, you said you worked on turtles, and you were, that guy living down by the river, or in this case, the lake.
Yep.
And what were turtles kind of like what driving passion for you then and now, or is that something that was kind of more opportunistic and something that kind of presented itself to you?
No, turtles have been a driving passion, since I had that one as a child. And this particular turtle I worked on was the eastern river cooter, a very rare and threatened and endangered species in Illinois. And they're hard to capture because they are primary herbivorous. So you're not going to get them in baited traps. You're going to get them snorkeling, or you're going to get them in passive traps. So I took a route I really wouldn't have recommended to graduate students, a very rare species that's difficult to get, and now go try to put together a master's degree on it, but
Yeah.
ended up doing it. Yeah.
So,
Yeah.
what was the focus of your thesis?
It was population and community or assemblage ecology at the freshwater turtle assemblage there at the site, with the emphasis of whatever population characteristics I could get from the eastern river cooter. Like I said, it's on the periphery of its range in Illinois, so it's of deep conservation concern, and we had very little information on it. The only things that were known about the species before I started was Dr. Mall had found one nest, and then just a handful of specimens in collections.
Yeah.
So,
Huh. Yeah, I never really thought about it, but I'm guessing Illinois is probably an interesting state in some ways. It's dominated by what would have mostly been tall grass prairie, but you have some, in the South, you've got kind of this whole Mississippi River drainage coming up, and probably a
yep.
lot of species that, are kind of ending their distribution in Illinois. And then you've also got the Great Lake right there, which adds another, kind of different level of diversity. So probably actually a pretty interesting state with these different ecoregion and water bodies coming together.
Yep.
So,
Yep. Southern Illinois is really really interesting. With the kind of the Ozark uplift and the coastal plain meeting there, and you get just on both sides of the state a really interesting mix of species. I mean, we have eastern mud turtles, as well as sirens and mud snakes. In Illinois, you go all the way, to the north, and it's Blanding's turtles, and line snakes. So it's a very very interesting, biogeographical region, pretty herp diverse.
Yeah, I was able. I won't give the details on locations, but I was able to go to a facility that I'm sure you're very familiar with and maybe work with. Let's just say in the greater Chicago area, that where there were heads starting and doing a lot of captive husbandry work with Blanding's turtles and restoring populations in that general region. And it was a pretty impressive project. So
yep, very familiar.
Yeah. Great. So you're doing the masters on this cooter. Is the cooter rare mainly because it's on the edge of its range there, or are there other factors that are kind of causing its decline across its range?
Yeah, you got generally it was probably likely very rare because it's in the northern periphery of its range, but then compound that all the channelization and leveeing that happened, throughout southern Illinois for cultivation and wetland draining. So you lost a lot of those wetlands associated with the major rivers, that would flood a lot of floodplain lakes and wetlands. So, a little bit of both, but we're now finding out, because they are difficult to capture and difficult to detect, that they're probably more widespread than what we thought.
Gotcha. So you're living on the edge of this lake, pretty much homeless.
Yeah.
Complete turtle bum at this point.
Exactly.
And what are kind of the next steps? How do you finish your master's and where do you go after that?
Well, I did want to go and do a PhD, and that's when Dr. Mall had kind of set up a few meetings with people that he knew in the field. Was Chris Phillips up at the Illinois Natural History Survey. He had just taken the job as curator of herps a few years before. There was, I met with, Justin Comden at SREL. one of my, probably greatest mentors in a few meetings, that I had with him. So he has had a lot of sage advice. So, went around and then kind of interviewed. I had wanted to continue on the river cooter and do a long-term study. So I'd always been fascinated with long-term studies. And of course, working with turtles, you need to study them in long-term to really get any information. And, when you tell people what long-term is in a turtle, 30, 40 years, they're like, what? I'm like, oh yeah, you haven't even made it one cohort generation yet.
Yeah, and Congdon is a great mentor for that, obviously.
Yeah. So. Yep. He had lots of sage advice how to do a long-term study and keep it going. But
so you go to these different interviews. How did it end up panning out for you?
So I could have went to SREL and could have went to and worked with Justin, and again worked with Chris, Dr. Phillips at the Natural History Survey. It boiled down to where I could effectively continue the River Cooter project. And at the Natural History Survey, it was, yep, you're right here in Illinois. We can continue to work on that. At the SREL, it'd be like, well, it'd be a bit of a stretch, to try to get funding a little bit more difficult. So,
what do you think about these chicken turtles?
Exactly. We found out lots of fascinating things about chicken turtles. Kurt did that. They might as well be the cricket frogs of the turtle world.
Yeah. But, Okay, so you stay in Illinois, continue to work in the same system, and so do you stay at the same university for the PhD, or do you go to?
Nope. I moved up to the University of Illinois and started to work on the River Cooter. It didn't quite go as well as planned, and that's just the way dissertations go, and it just ended up in. A turtle PhD wasn't in the cards for me. But what had presented itself was this study on the eastern Massasauga rattlesnake at a site where they thought that they knew they had them, and there was some sort of a development, a resort was going to go in, and it could have impacted the snake.
Yeah. So, Let's hold off on talking about that Massasauga project in detail, because I do want to talk quite a bit about that in the here coming up in the main part of the episode. But so you do your PhD on Massasaugas. How do you end up getting from there to where you're sitting today, and where are you sitting today?
Oh man. So right now I'm sitting as kind of the director for the Population Community Ecology Lab at the Illinois Natural History Survey. And on Friday, you and I just kind of briefly talked about it before, I will be stepping into the role as interim director for the Illinois Natural History Survey. Time and lots of work and lots of connections is kind of, what got me, and a lot of collaborations, a lot of good people working with, what got me from finishing the PhD to slowly building where I'm at and kind of building a program.
Yeah. And you were working with that group on your PhD, correct? You were talking with Chris Phillips and
yes. Yep. Chris was my doctoral advisor. Yeah. So, yeah.
Okay. Gotcha. And is he still there? Where is he?
He's retired now. So Yep. So he still does some herp work, and we're still on some manuscripts with him that are floating around and stuff.
Gotcha. Okay. I think I met him a long time ago when we had the snake ecology group meetings there in Illinois. Yeah. How long? That was 20 years ago. A long time ago. I don't know more than that.
Touch of nature. Yeah. Yep.
Yeah. But anyways, great. So you're working your way up, like you said, time, dedication, just this long-term view. And you've really, in my mind, when I think of like herpetology and just really ecology in general in Illinois, you're who I think of. And so you've built this entire career around working primarily in one state. Which is fascinating. A lot of people do the exact opposite. They bounce all around, and either strategy can be successful. But do you have any thoughts on that relative to if somebody was like you and they were dedicated to a particular place and they wanted to stay in that place, and there were kind of forces trying to pull them, to work on chicken turtles in South Carolina or whatever it might be. What, just any thoughts on that kind of approach to a career trajectory?
No, I mean, for me, it was all just kind of maintaining a long-term vision of what I did, and I can be pretty stubborn. When I started off, doing the work, let's say on it, I know you want to talk about the Eastern Massasauga a bit later, but I wanted to see the project to fruition. Mm-hmm. So that's what kind of kept me cemented here. I knew it was going to take long-term dedication for it. You can go either way. I mean, here it's just been working with the land management agencies, working with the forest preserve districts and the DNR, repeatedly, and building kind of a reputation with them, and then building a good, cohesive, collaborative, group here. Like we talked about, Illinois is pretty herp diverse. So there's no shortage of work, conservation related work to do. I do venture out, as well, mainly in more of a kind of in a collaborative role. People, I guess, would see the type of data analysis I do, and then come to me, asking for things, bouncing around, great. I mean, like one of the sage words that, Justin had me do was read Don Tinkle's paper on the importance of long-term studies. And, we're kind of built to have to bounce around. You've got a limited time of master's degree, you got a limited time in a PhD, you got a limited time in postdocs, right? Mm-hmm. And then you might bounce around for a couple of jobs, before you get settled and tenured, and then tenured, you can kind of think about things. That is, as I think, how the system is built, and people like Justin and stuff can kind of buck the system as well. And I think I bucked it a little bit too by being able to maintain a long-term vision. Either work, and people, people are successful. I'm just, I just like to be personally, if you're a person that has a long-term vision and stuff, it would probably behoove you to kind of dig down and make the best of, what you have,
Yeah. Along those lines of long-term. So are you currently able to continue this long-term work on the cooter that you envisioned, even though that project didn't come together for a PhD, or?
I did. Yeah. And I was still working on it a little bit, trying to get it jump started while I was doing my PhD on the Massasauga. But eventually I had to kind of give it up,
Gotcha.
And that was pretty hard. I'm sure there are still mark turtles out there. I have always thought to myself, maybe I'll try to get a grant or contract at the end of my career to do the river cooter work again, and just kind of end where I began. And I think it would be a good full circle. But sadly, that one had to go by the wayside. But we do have a bunch of other long-term projects that are ongoing.
So, what part of Illinois are you based in?
I'm based in Champaign-Urbana, so right there in central Illinois. Okay, East Central Illinois.
East Central, yeah.
Yeah.
And so, I mean, Illinois, we're talking about as a state. The southern part of Illinois actually has quite a bit of public land, and that's where you get this really kind of interesting, almost like we'll call it southern diversity, coming up into a state that goes pretty far north. But a lot of the state of Illinois is dominated by private land. And while I'm assuming you do some work down south, I'm assuming a lot of the work that you've done over the years is working with private landowners. How has that been throughout your career?
Surprisingly not. We don't work with very, very many private landowners. I'm mostly working on public lands, whether it be from Forest Preserve Districts, Nature Preserve Commission, the Nature Conservancy, Audubon, Fish and Wildlife Service. There is, as you brought up, quite a lot of public land still in Illinois and throughout. The private land stuff, once you get up into central Illinois, most of it is agriculture.
Mm-hmm.
So there's and what is still native is mostly protected already. So I have done very, very little actual private lands work.
Oh, interesting. Okay.
Yep.
Well, as I mentioned in the intro, we're going to kind of talk about a number of different projects, but I want to kick it off with the long-term Eastern Massasauga work that you've been doing. This is the Snake Talk podcast, and we certainly will talk more turtles, but I do want to talk about Massasauga. Is certainly one of the most fascinating snakes we have here in North America, and certainly one of the most threatened. So I guess let's start there. If we look at Illinois as a state, like maybe describe the status of Eastern Massasaugas and maybe how that's changed over time.
So they probably occupied the northern 2/3 of Illinois pre-settlement times, and there are, records of people encountering dozens in a day. There's records, over by the St. Louis area of people collecting them by the buckets, and even over at Allerton, near us, there are records along the Sangamon River, historic ones in numbers. They pretty much faced what looks like the best to us a severe decline back in the 70s, likely associated with the removal of secondary grasslands, right? So they could inhabit the native wet grasses, wet prairies and stuff until those, I guess, were turned into hay fields, right? And you had smaller fields and stuff like that, but those secondary grasslands, they were still in pastures. They were still able to occupy, right? And those, there was a lot less smaller scale agriculture. Then you're going, through the 50s, 60s, and 70s when it starts to become large scale mechanized agriculture, and we lose the secondary grasslands. So the decline of the Massasauga mirrors a lot of the decline of the grassland birds that we've had in Illinois as well.
So when you
with the losses, yeah.
Sorry to interrupt, but when you say a secondary grassland, are you saying like these are grasslands that historically they wouldn't farm because they were a little wet, they were, the edge of a field, they were a corner, whatever it might be? What is a secondary grassland?
I'm more of like a pasture, if you think about it, or like a hay, hay type field. Yeah.
So not a native tall grass prairie, but some grass in there that they're grazing. The grass might not even be native, but the snakes are able to persist.
Exactly. Yeah.
And then you have large scale agriculture and just kind of farms getting larger and larger, and they're probably filling wetlands and farming right to the edge of the rows and on and on.
Ditching and you name it, drying out the habitat. And so then we see in the 70s another decline. We get a State Endangered Species Act in the early 70s, and the Eastern Massasauga is one of the early species that is put on the act as listed. By the 70s, maybe we had, I'm trying to think offhand, maybe 10 or so 10 or so populations, or 12 left, and then just over time those have dwindled to one or two that we know about now.
Yep.
So pretty catastrophic, pretty severe decline, and surprisingly, Eastern Massasaugas are not inhabiting anything special, right? So it's not like they're inhabiting high quality, wet grasslands and everything. What they're inhabiting, you can find all over Illinois, but why are they gone? Why are they gone from these sites? Yeah.
So even though you've had these changes in agriculture and there's been more habitat wetland loss, there's still a fair amount of abundant habitat that they could survive in. They're just not there.
Just not there. Yeah. You'll see these wet fallowed fields all over Illinois, and you'll think to yourself, oh yeah, Massasauga habitat there, but why are they not there anymore?
Yeah.
Is it just now a historic thing that that field may have had them in its history, but it's been on and off farmed, for 50 years, and that's just depleted populations nearby.
Yeah, and for them to
Yep.
recolonize areas is probably pretty difficult in that
It
way.
is extremely difficult if you are not if you're not restoring habitat adjacent to known populations. Forget it. They're pretty site faithful. They don't move very far, so they almost have to expand out from the center.
Yeah. And what's their life history like? Meaning, I'm really kind of getting at their reproduction. Like a timber rattlesnake would be an extreme example. Late maturity, multiple years between pregnancies, on and on. What is their life history like relative? Because that obviously would have a big impact on, their ability to recolonize.
Yep, it is variable within their range. Southern in Illinois, where we do work. Females can start to reproduce. They can mate at age 2 and reproduce at age 3. So amazing capacity for population recovery and maintenance. So I think the largest litter size may have been 21 recorded from an Illinois population. Tom Anton kind of studied that. I would need to look up to see that reference. But as with many species, the further north you grade, their life history changes, particularly for ectotherms. So Massasaugas at their northern range limit are going to be slower to mature. They're going to have a longer time likely in between reproductive bouts. Surprisingly enough, they're not maturing or getting to a larger body size. Our Massasaugas that we study at the remaining site are actually slightly larger in some cases than those at the northern range limit. And we think that that's pretty much a resource-based issue. Massasaugas at that site are able to capitalize on mammalian prey right after birth. So, as neonates, the southern short-tailed shrew ranges up to where this population is. So Massasaugas are able to consume energetically nutritious prey right off the bat. Whereas in other places, they might be, consuming other snakes, consuming lizards, taking a little bit more time and not getting that high, kilocalorie, punch that mammals give. With fats and everything. So lots of life history differences, faster growth rates, that we see, at our study site in Illinois versus, range wide. We see longer activity seasons. So all of this feeding into larger snakes with more energy, more offspring, better offspring, quick time to recovery.
Reptiles face many challenges, from habitat loss to climate change. As an example, turtles are the most threatened group of animals on the planet, with over 60% of species classified as endangered. The Orianne Society is working hard to secure a thriving future for these incredible animals. To engage in the critical work we're doing, explore our website at www.orianne.org. There you'll find in-depth insights into the science, conservation action, and communication initiatives we're undertaking. Visit us today.
Yeah. And you mentioned, we kind of started this life history-oriented part of the conversation. You were talking about habitat that you see that you're like, oh, that's Massasauga habitat, but you don't find them. Are there any thoughts on other factors, maybe like something more like insidious, like a disease or, I know, for example, snake fungal disease, Massasaugas are one of the species where it's, there's been decent prevalence. Correct me if I'm wrong. Anything like that, some invasive species, anything else that might explain that pattern you're seeing of available habitat, but no snakes?
So we've done Ophidiomyces work at our Illinois site and found its prevalence goes all the way back to when we started the study. So it may be a native, a native fungus on the landscape, similarly to the way the North American chytrid fungus is. So this Ophidiomyces just may have always been there. There are lots of other, insidious things. Rattlesnakes are called vermin, and all the way up through the 70s in the Chicago region, Boy Scouts still used to have rattlesnake roundups around one of the sites, and they used to,
get the rattlesnake killing patch.
The rattlesnakes, get the rattlesnakes from the one or two patches that were left to kind of, wipe them out, to clear the landscape of vermin, so to speak. Yeah.
Mm-hmm. Yeah. What you guys have Horridus in southern part of the state. What's their status in Illinois?
Well, we have Horridus in the southern part of the state, west central, pretty much, ranging up the bluff lines of the Mississippi River, too, as well, and they are state protected as well. They are listed.
So talking back to the Massasauga, so you, as you mentioned, there's a state level protection, but they also received federal protection recently as well. Do you want to maybe touch on that? And has that changed things in Illinois for the better or worse?
We were doing, I think, pretty good with the conservation of what we had left in Illinois, and for the federal status, they sat as a candidate species for a very, very long time, and then were finally listed. The Western Management Unit, which encompasses Wisconsin, Illinois, and Iowa, is doing the worst, out of the groups. Although Wisconsin, they're starting to find some good populations and some, relatively healthy and robust populations. Illinois and Iowa, we're not doing so hot. So I don't think it has had. I mean, there's not much you can do. We have one or two populations left. So, beyond that, we could afford it all the levels of protection, that we want, but they're already on protected land. Mm-hmm. So, it's more beneficial, I think, to other portions, of their range. It does open up some other, potential conservation dollars and things like that, but those are pretty limited. The more species you put on a list, it doesn't mean the more money is allocated. It just means you're divvying up the pot more.
Exactly.
So to speak. Yeah.
Well, I do want to get into the obvious, conservation approach, given you have a lot of habitat, but not a lot of populations, and the concept of potentially trying to restore populations. But I want to save that, just thinking about the chronology of our Massasauga discussion. I want to go back and talk about your PhD work and your, initiating this long-term work. So as you were going into, I'm assuming you're working at the same site that is the remaining site. I think there was a second site that was pretty significant even 20 or 30 years ago, and that's blinked out.
That has blinked out.
And so tell me a little bit about your dissertation work, kind of going in, setting up this long-term work. What were some of the major questions you were interested in and all of that?
Yeah. So I said I wanted to see this to fruition. And then one of the things that Justin had instilled in me, is if you want to do a long-term study, you've got to think about, shorter term questions to answer along the way. Otherwise you'll never get any publications, and you won't get hired anywhere. You won't do anything. So have the small questions, but the long-term vision. So my long-term vision was always more of a life history type approach to it on one hand. I always wanted to see if I read Williams's Adaptation and Natural Selection, and one statement in that book really struck me, that organisms should grow fast through periods of high mortality. So I should see a relationship between the growth rates in cohorts and the survival in cohorts, right? So we started this Eastern Massasauga study back in 1999. I now have 25 data points, where I could potentially do that. So that's the thing. That's how long it's going to take to develop this progression, right? So I can ask very, very few questions. But, so that's the science part of it. I really want to, kind of answer something like that. And Then there's the other long-term vision, which is the conservation and the recovery, right? I said I wanted to see the study to fruition. Well, fruition either means they become extirpated or we have recovered them and stabilized the decline at the sites. And naive Dres starting his PhD, didn't realize that until Dres was mid-career and went, holy crap, I'm going to be at this until I die. Hopefully, because that means they're still on the ground, right? So it started there, and the initial work was, basically we're at the southern range limit for this species, and we need to know everything that we can. So it became a very holistic approach to the study as well, answering, in successive refinement, other questions. And I had a good pair of people that were working with me. Dr. Ben Jellin, who, did his master's degree there, started off as a technician on the Massasauga project, and Dr. Don Shepard, who started off as a technician there. And the three of us would just read papers and just, dream up things and then just add to the data sheet, right? And add to the data sheet. Just simple things, even check boxes that, 25 years down the road, we have a crazy amount of data now. We took blood from every snake. So we could look at like allele frequencies and behavior, of an individual snake. And it just, you name it, the relationships of the data that we have now. But it was, to drive at three questions that you really have for conservation that I try to drill into my students. It's one, where are they? Where do we have them on the landscape? How many do we have? And what habitat are we using? And that pretty much was the foundation of my dissertation there, using, demographic work, using, radiotelemetry work for habitat, and using our visual encounter surveys of the habitat to find out what patches they were at.
Yeah. So answering some of those questions, like, first of all, in this remaining robust, population, how many are there? What kind of, maybe like population estimates, survival, any other kind of demographic, estimates that you found? What did you find in the population there?
Lots and lots. Yeah. So, we've got some long-term stuff that, the population has fluctuated over the years, but there's only maybe about 400 or 500 Eastern Massasaugas on the ground kind of at any one time. At these sites, there were periods where it was really high, and then we can then attest, like there were periods where there was no management happening, that we saw declines, in associated population sizes. Basically, right now, the highest populations that we estimated were when we first started in about 2001. The snake populations were booming. They declined, and we're right about back where we started. So 26 years later, there's been kind of a full cycle where, they were doing great to then, people can attest, over the years where Dres is out in the field and we're not catching Massasaugas, and then he gets nervous because when you see populations decline like that, it could always be the next year you go out there and you don't find them anymore.
Yeah. Not just populations, the population too.
Exactly. Yeah. So and we've seen that with a couple of three Massasauga populations in Illinois. You're finding them, and then that's it. They hit a critically low number, and then blink finally out.
Yeah. So can you speculate, or maybe you don't have to speculate, but what that pattern that you've observed over time, a population decline and a population increase, is there anything you can speculate as to why that's occurred?
The only thing that it has really associated with was amount and intensity of habitat management and proper habitat management. So the more years that the habitat goes unmanaged and invasives take over, those are the years that we see the decline, and it's likely a cascading effect with prey bases and prey abundances. So
what does Massasauga habitat management look like? Is it prescribed fire of these prairie wetlands? Is it like mechanically removing shrubs? What is management of Massasauga
I'm going to
habitat?
say yes. It is. Yeah. It's doing prescribed fire to keep woody vegetation from encroachment, to keep it in that early seral stages. It's mechanical removal. We have autumn olive and Russian olive, at the sites, and those readily take over open canopied habitats pretty quickly. It's even chemical spraying and herbiciding to remove invasives. The big issue that we have now is the Sericea lespedeza, and it will just take over everything. And
what's the common name of that?
I don't even know what the common name of it is. I think Sericea is actually the common name.
Is it like a grass, or is it?
Yeah, it is. It is. Let me look it up real quick. But I think.
So there was a time period where these habitats weren't being managed, and then you guys in recent years have ramped that back up.
Well, it goes through ebbs and flows. So I mean, over a 26-year study, right? There's only a few of us in the agencies that have been here the whole time. Kind of the old guard, and people kind of come and go in their career. Over a long term like that. I mean, we've studied it almost an entire career, 20 years if you think for some people. Different priorities, shifting priorities, different species coming in, different levels of funding. It's always a challenge to try to save these things long term, save these habitats and manage them in the long term. But here we got more energy again, here on year 25, 26, with some.
Any from that long-term data set, kind of any interesting superlatives, meaning like an adult snake you found 25 years ago that, you found alive, or any like longevity, or just anything like that, interesting data points from a long-term data set?
Yeah, I think the oldest individual that we have would have been at least 13 years old, and it was a male.
And what is longevity for a Massasauga thought to be?
That's a good question.
I don't know.
It comes. I'd have to go to the, what is it, Snyder and Bowell, the longevity and herps. I think it was.
It certainly would be much less than something like a timber rattlesnake.
Yes, it's particularly at the southern range limit. The Massasaugas here have about let's say 70 to 80% survival rate, and even less if you're a female. So that you think 80% is good, but it adds up, pretty quickly, and you can get knocked out of the population. Yeah. So this male was he was an adult when we captured him. So, we're saying at least 13. He could have been older. Mm-hmm. Without doing some skeletal chronology, which we do have a lot of Massasaugas in our collection that we've collected from roadkills or other mortalities that we've seen. We could, do some sectioning of vertebrae and see how old the snakes are getting as well. We have some older females, that may have had, 2 or 3 reproductions, but I just said that females have, a higher mortality rate. That's common in the pit vipers. It's just so energetically costly for a female to reproduce. It becomes difficult for them to recoup those resources before, they've got to go back into an overwintering state. They just have a very narrow window to do that. So, it's almost what, 50%. It's almost a coin flip, whether they're going to survive or die. So, for a female to reproduce 2 or 3 times is amazing.
Yeah.
And that's one of the caveats we found with doing our long-term data analysis and life history analysis, that the population growth rate is highly dependent on primiparous females. And that makes sense. Yeah. Most times that may be the only reproduction that they get, that they can get off in their lifetime. And then everything else, it does add, second and third reproduction would add to population growth rate, but not as much of a component, to that. Yeah. Yeah.
Well, so you talked about kind kind of this long-term approach, the life history approach to this studying this snake. But you also talked about that idea of putting together small projects on the side that kind of
Yep.
piece together a bigger picture. We don't have time to go through all of them, but any really interesting kind of smaller projects that are part of this long-term data set that you think is interesting and the audience might take interest in?
I could go over lots of them. I find them all interesting as I do. So some of the early work that we did on diet, palpating individuals to see what they were eating, and then just a little.
And that's where you learned that they were eating more mammals, throughout their life than, say, other populations.
Yep, exactly. When we would palpate, these juveniles in the field. It would most likely be the southern short-tailed shrew. There were a few cases of what we call gluttony, where they were able to swallow an adult deer mouse, but we think they compressed their lungs and killed themselves. It's like, because they were so distended, these snakes, and it was like, my gosh, if they were able to pull that off, they'd be set for a year or two. But it was very, very risky. We had so many neonates 1 year too. We just did a dietary preference study to see what they would eat, because the literature said they'll eat Orthoptera, grasshoppers, they'll eat, they'll eat frogs, they'll eat other snakes. Okay, we'll see what these neonates actually eat. And there was preference for mammals and a gait limitation associated with it, because we fed them pinky mice. All snakes offered a snake, pretty much ate a snake, perfect thing. Only one offered ate a frog, but in other portions of the range, they will eat northern leopard frogs and other frogs. None of them were interested in any arthropods, whatsoever. But we know Trigeminus, will eat arthropods and centipedes. Mm-hmm. Yep. So that was a very interesting thing. Another big one was. It became Ben's thesis, which was just looking at the mating system and the sequence of mating, and it was a little bit of both. It was a little bit of mate guarding going on, and a little bit of kind of a scramble competition type thing. So it was on a continuum. Heavier males tended to displace lighter males, according as a suitor male as the season progressed. So that was kind of interesting. Dr. Sarah Baker did some good work on reproduction, a little bit more on demography, and one of the first kind of viability analysis. But I think a lot of her big work was working with Matt Allender on snake fungal disease in the population. Is that they're the ones that turned in. She's the one that turned in first snake with snake fungal disease. And
first Massasauga or first snake?
The first Massasauga that, in the population. And it was like, we've seen this before. We saw some kind of this cauliflower head before when I was doing the work, and snake was in bad shape, held on to it. And then, the rest is history, there. So lots of side stuff, too. Lots of genetics work, and we've partnered with a lot of other collaborators on things. Another one that took off, and I really like this one, is we put together Massasauga movements with turtle movements to ask the question about road crossings. Right. So we had a bunch of telemetry for turtles there that Andrew Coons, Stealth and Natural History Survey, was doing there for his master's degree, and our radiotelemetry for snakes. And we just did a simple, kind of random walk analysis. Predicted how many times a Massasauga, let's say out of, I forget, any analysis, out of 10,000, random walk paths would cross a road, versus how many times a snake, actually did, and found that there was some road avoidance going on there. Snakes crossed roads less than observed, and it was most stark, with snakes that were using ditches, moving up and down ditches. They would just stay on the one side of the road and not cross. This countered kind of, you would think, some of the work that we did with road mortality around the lake. We drove the same routes every day. So we scraped up every piece of road bacon, for herbs, along there. And we found a lot of Massasauga roadkills. So that really kind of led us to believe, how do you rectify the two? Man, they're dead all over the roads here. Well, road crossing is a rare event, but when it happens,
it's deadly.
It's high. It is deadly. It is deadly for the snake. So, and that paper that we did with the random walks, I think we wrote that paper in 2 weeks, analyzed the data and everything, and it went to animal conservation, and it's highly cited now.
Yeah,
it's amazing. Yeah, I mean, I get Google alerts every now and then, to see where it's being cited. They're citing it with primates, and I'm like, oh my goodness. I'm like, we've made it to like introductions and stuff like that. This isn't. Yeah, so.
We've saw similar patterns with both lutosis and horridus in different places where, these animals, they're going right up to the roads, and their home ranges are perfectly on the edge of roads. And then, but then we do, so all of the animals we track, we're never having them cross roads. But again, like you saw with the Massasaugas, we do find roadkills. So that's interesting. But, anyways, well, let's talk a little bit. We talked about the status at the state level, but then also at the federal level. Let's talk a little bit about the other component here outside of the research, and that's the conservation and recovery. I'm not familiar with the recovery plan for Eastern Massasauga, so I don't know what all that entails, like what kind of your goals are within Illinois to reach recovery. But what's kind of, I guess, just high level, like what's required for recovery or to take this animal off the endangered species list, and what are some of the strategies you guys are using? And at some point, I mean, talk about that husbandry idea and the idea of kind of repatriating populations and some of that.
If you enjoy listening to the Snake Talk podcast, stay in the loop with the Orianne Society's latest conservation updates from the field. Read about new published research, as well as fascinating insights on the species we work with by following us on social media. Follow, like, share. Let's spread the word. Together, we can make a difference. Find the Orianne Society on Instagram, Facebook, X, or LinkedIn and be part of the conversation.
Yeah, so this is a tough, call here. And now studying the species for many years and thinking that, having all the energy, in the world, but just seeing the decline and what it's going to take. I would honestly have to say that the Eastern Massasauga is not recoverable in Illinois, and that the best we can do is halt the decline and maintain what we have. Just the number of populations that have declined, the level of effort that it would need to take to repatriate these populations, right? So, and where these populations would need to be repatriated. Can you imagine, having a program of releasing venomous snakes into, urban parks and stuff where the species used to occur? That is not even socially feasible anymore. So the best thing, I think, and we did studies, on. And there have been other studies about repatriating the snakes. You would just need so many snakes to drop into a habitat to get them to take. Now imagine, snakes having, let's say, even neonates having a 50% survival rate naturally. That's with them choosing their hibernacula that they know that they can survive in. How about us choosing the hibernacula for them? That first year survival rate is going to go, into the toilet, especially if it's in a case where they couldn't even scent trail, conspecifics to a successful hibernacula, an active crayfish burrow.
How about
we modeled? You would need hundreds and hundreds of snakes a year, and no facility can really produce that.
You mentioned, so given that's the case in Illinois, so you've got the one real strong population. You said 400 or 500 snakes. How and you mentioned genetics in terms of genetic diversity, and then maybe long term thinking. Is there any kind of like assisted migration that's being planned across? Because a lot of Massasauga populations would have the same issue. They'd be very isolated.
Yep.
So, is there any kind of part of the recovery plan that's like keeping genetic diversity up by making, say an individual movement or breeding snakes and
Yeah. So right now, using microsats, we haven't really detected any bottlenecks or any reduction in alleles. However, using conservation genomics, people around the range are starting to find out that microsatellites were kind of hiding that there is inbreeding depression, that there is these issues when looking at the whole genome. So we will start looking. We're putting together a CSWIG, with the Western subunit that will look at conservation genomics of the species using as many of the samples as we have banked over since 1999. So we'll be able to hopefully answer, with the latest techniques, if those are, necessary, right? What's the effective population sizes? Do we have inbreeding? Did we hit a bottleneck, more, across the entire genome rather than just, these neutral alleles? Mm-hmm. So could be, right? Right now, within the region where we work, there are 3 isolated genetic demographic units.
Gotcha.
So how strong those are, genetically and demographically is kind of another question. One is really strong. The other 2 are kind of weaker. And it makes sense that these, that there are these units even within the relatively small landscape that we're working on. They found up in Canada, along the Bruce Peninsula, that what was it, even 7 kilometers distance, there was genetic distinction,
Gotcha.
among populations. So that goes back to, you have to build around them because they are very, they have high site fidelity and limited vigility.
Mm-hmm. So maintaining their population in Illinois then is largely monitoring whatever demographics, genetic diversity, different components, and then sounds like habitat management is an important component. And then, so how about if we zoom out to range wide? I don't know if you probably don't want to speak for other states necessarily, but like in terms of recovery, I mean, do you think this is an unrecoverable species at the federal level, or that it's going to need to be maintained on the endangered species list, and we're going to just kind of need to maintain populations where they are? Do you think we can recover it?
It may be. It just may be on life support, but Canada and Michigan, do have some of the largest, more robust populations. But then when you look throughout the rest of the species' range in New York and Pennsylvania and Ohio, Indiana, Illinois, and Iowa, it's maybe 3 or 4 populations at most, per state, and varying abundances. Wisconsin, like I said, is finding more, than I thought. I think we're pretty much maybe extirpated from Minnesota, and so maybe Wisconsin will be able to help bolster numbers with what they're finding. But definitely anything outside of those, the Great Lakes area, Wisconsin, Michigan, and Canada. Mm-hmm. And even a couple of the Canadian populations are kind of in bad shape and kind of on life support. Mm-hmm. So
last Massasauga question, then we'll move on. But and I kind of know the answer, suspect I know the answer to this, but I just think it'd be interesting for the audience. I don't know how much experience you have with like the desert Massasauga, but what makes the Eastern Massasauga so different as compared to the desert Massasauga and so much more threatened and rare?
You know, what do we really know about the desert Massasauga? I mean, you might want to talk with Andy. I think you have talked with him. They may be in just as bad shape, if not worse.
Yeah,
than the eastern Massasauga, and there's concern about Tergeminus too.
Mm-hmm. Yeah.
Range wide, are they declining? But Tergeminus was thought, hey man, the species is everywhere. It's pretty common, but now the more you look, maybe they're not as common anymore. There is some concern. But desert Massasaugas, well, they're while not being as genetically distinct, we'll say, because there's question as to how distinct the subspecies is. Mm-hmm. But population wise, I don't think they're in very good shape either.
It's just that we don't know as much about them.
Yeah.
I mean, it seems like the landscapes that they inhabit, impacted, are less impacted than much of the range of the eastern Massasauga.
Yep.
So, but, I started off this episode talking about how we were going to just discuss all these different projects that you're working on, and we just spent the whole episode talking about Massasaugas, which I'm not upset about at all, because I think they're such a cool snake.
And I've spent 26 years talking about Massasaugas, so
and so maybe we'll have you back on one of these days and talk about some of these other projects. Yeah. But I think we'll kind of wrap up the body of the episode for there. I think it's right there, because I think that's just an incredible amount of information on Massasaugas, and people are going to be really interested. So, as you know, before I let my guests go, I like to have each of them tell me a good snake story. So, do you have a snake story for us?
Oh, I do. I do have a snake story for you. So, and it involves my Massasauga bite. So, generally it's not good to come up to somebody who studies, venomous snakes and immediately go, "So have you been bitten?" Answer is likely yes.
Not me.
It's not necessarily a badge of honor. So back in the day, we, weren't as safe as we are now, with working with the snakes. And we used to record substrate temperatures, right, with an old dial rheostat thermometer that you would stick, and you'd want to get it as close to the snake as possible, right? Because we had temperature sensitive transmitters, and we want to get the substrate, as close to the snake as possible for any just little microclimates. You want to capture that. We are out in August. So if everybody knows snakes in August and everything, they are jazzed up on the heat. They're ready to go. Their physiology is firing on all 8 cylinders there. It's during the mating season. The particular snake was shedding. So you see what's happening is we're setting up a whole group of errors here. And we had high school teachers shadowing us for their continuing education.
Oh, no.
Yeah.
Of all the days, that's great. Well, it's not great.
Yep. So we get out there, the day's going great. We're tracking snakes and everything. And we come up to this snake who's been basking, thermoregulating in preparation for ecdysis. It can't see, but I'm sure it could sense the entire herd of people, I've heard there were 6 of us come up to surround it. I reached down to put the rheostat temperature probe into the ground, was right within the strike zone. And just like that, snap of a finger, pow, pulled back, everything, and I knew I got bit. So
what could you feel that? I mean, it's obviously it was very.
I felt the snake hit me. Yep, and then I was like, yeah. I won't use the profanity here, but there was in the head, oh crap. So I pulled back, and Ben Jelen was there, and he looked at me and he goes, "Did he get you?" The high school teachers didn't see anything, and I held up my hand, and at my right hand and my thumb, the blood was just already dripping down. And Ben's eyes got big, and then Don Shepard looked over at me, and he's like, "What the?" And I'm like, we don't want to panic the high school teachers. So I got them aside and I said, okay, this is what we're going to do. We're going to take me back to the trailer because we were living in a 1970s mobile home, on site. So I think maybe you've seen pictures of that on my Facebook. So still almost kind of homeless while doing my PhD. I said, take me back and, you guys continue on with the high school teachers, finish the rest of the snakes at the next site. And they're like, well, what are you going to do? And I'm like, and they're like, the thing was, who's going to call Chris? And I'm like, I'll call Chris and let him know. And just like take me back and tell him I got to do something else. So they did that and then, went off and I called Chris and he immediately was, you guys have to stop doing this, pulling pranks on me. He goes, I can't sleep as it is, knowing you three are down there handling venomous snakes all the time. And I'm like, no, this is true. I was bit, and immediately it was, all right, how are we going to handle it and everything? And I'm like, if I show any symptoms, I will go to the hospital because there is a chance it was just a dry bite. Let's not rush to the hospital, get me pumped with antivenom and then I can go into anaphylaxis, or anything like that. Let's see what's going to happen first. And, so he's like, so what are you going to do? I go, well, when I get stressed out, I take naps. I'm like, so I'm just going to take a nap and kind of calm down here and see. Well, Don and Ben came back and, they called Chris back and Chris is like, is he breathing? Make sure he's breathing. And I snore and they're like, oh, he's snoring away. So yeah, he's fine. It turned out that, I don't know if I got a dry bite or not, but the snake did hit me with one fang and the fang went through the meat of my thumb, went in and out.
Oh, wow.
So there were two holes, from where the fang hit and went out. So I don't know if, he injected, but, the fang was out and he shot most of the venom out or it was a dry bite. I had no ill effects or anything like that. But the whole story is, it's like, oh my gosh, he got bit and he went to sleep. He took a nap. I'm like, well, what are you going to do? They say calm down. How can you get any more calm than to just,
I was going to say, you got a good nap out of the deal.
Yeah, I did get a good nap out of the deal. I didn't have to track the snakes the rest of the day. Right? I got out of that. So
that's funny. And I love how you hid it from the high school teachers. Did you guys ever end up telling them after the fact?
No. Yeah, we didn't. Yeah, we just kind of slinked it off and kind of went away. We didn't want to panic them and ruin their day and everything because they were having a good day and just like, we'll handle this elsewhere. This was one of those things that, not everybody needs to know.
Yeah. Great. Well, I love it. It's a great story. I really appreciate you joining us today, and I hope this new chapter with you taking over the interim director is a good one for you, and look forward to talking to you again soon.
Okay, thanks a bunch.
Okay, and I just wanted to thank the audience and tell everybody to remember snakes are animals too, and it's a privilege to see one in the wild.